Okay, so I think we can get started. So welcome to the third day of this workshop. And today I think we are going to be talking about challenges in omics analysis related to human disease. And we start our first speaker with Ron Shamir from Tel Aviv University that will be talking about the multi-omics integration.
Thank you. A common challenge that we all share is to make medicine more personalized. So while currently we use the same drug for all, and some are helped, some are, many are typically unaffected, and some are even damaged. We want to use additional data, omics, EMRs, etc., in order to tailor a disease that so that each individual will be helped. So I'm going to talk about 3 or 4 stories, and as this will be short, I will not be very technical. So the first story is a work of Nemo, a PhD student in the group. And the motivation is TCGA-like datasets where we have multiple layers, multiple modalities of data for a cohort of individuals, which is potentially extremely powerful. We have tens of thousands of samples across dozens of cancer types and 6 or 7 omics. However, these omics are extremely different in terms of their number of features, statistical properties, etc. Some of them are even not numerical. And the question is, can we use these together in order to, in our case, identify subtypes of cancer better? And we looked at the literature, and actually there are quite a large variety of methods for multi-omic clustering. I won't get into the different types, but here are a few representatives for each of the types. We even noticed that there is a subfield of machine learning called multi-view methods, which essentially does the same, but it was applied for other contexts, and many of these algorithms were not tested on genomic data at all. So we set out to benchmark these methods on 10 TCGA datasets, each of them containing a few hundred samples, a few hundred patients. And we focused on 3 omics: gene expression, microRNA expression, and DNA methylation. And we set aside the clinical data. There are about a few dozen clinical parameters available for each patient, and we put them aside for evaluation. And the evaluation was done in 2 ways. First of all, since we had the survival information of each individual, we could look at the survival curves of the different clusters after we clustered the samples, and use the log-rank test to assess the difference, significance of the difference between the clusters. And there were also about 6 clinical parameters that were common to all these cancer types, and we looked at the significance of the appropriate test between the clusters. I'll show you the results soon, because while doing this benchmark, we realized that maybe we can do better, and we came up with a new network-based approach. So here is the setup. We look at a particular omic and at the profile of a particular patient. And then we want to compute the similarity of 2 patients in this omic And we do this by this radial basis function, where it is normalized by this factor, which is the average of the similarity of sample I to its neighbors, where we focus on the k nearest neighbors. This is the eta that we see here, the similarity of j to its neighbors, and the overall the similarity between the two samples. And the relative similarity between two samples is this function, but it is normalized by the total similarity to the neighbors for j and for I. So, and the integration step, so this is done separately for each omic, and the integration step is the simplest possible, is just averaging this relative similarity across the omics. So here's the overall algorithm. We compute the similarity as we just described, and then apply spectral clustering. To determine the number of clusters, the only twist that we do is, rather than take the eigengap method, which orders the eigenvalues and looks at their differences and picks the largest, we multiply this by I, the index of the difference, in order to nudge the number of clusters slightly higher. And note that this approach can directly address missing data with no need to imputation, because we just sum over the average over the omics that are available for each pair of patients. Okay, here are the results. So we compare this on 10 cancer types, and compare the method that I showed you before, and Nemo is in the last column. So here in the first row, you see the number of cancers for which there was significant different survival between the survival curves using the log-rank test, and Nemo had the highest number. When we look at the number of omics where there was a significant clinical enrichment in one or more of the parameters, we see that Nemo is best, tied with 3 other methods. In terms of the number of clusters, it's sort of in the middle. There are good methods that create a large number of clusters, others do well with less. And in terms of runtime, it is the second fastest. The only faster is spectral, because we use the spectral algorithm as a subroutine. Here is another view of the results. So here, rather than counting the number of significant values, we summarize on the x-axis the log of the log-rank p-value, and on the y-axis the average number of significant clinical parameters, again averaged over the 10 cancer types. And we see that Nemo is on the Pareto optimal contour together with the RMKL and MCCA. So there's no absolute winner across all methods, but Nemo does well. Here is one specific example. This is AML, a cohort of nearly 200 patients, the same 3 omics. There was full data for only 170 patients. So what you see here is the survival curves for the 5 clusters that Nemo found. And here Nemo performed better than all the other algorithms, better than using just the 170 samples with the full data, and better than any single omic separately. And when we compare the results to the French-American board classification of AML and to our known subtypes APL, we both got a good match. So to summarize this part, we performed the benchmark of multi-omic clustering. Some multi-view methods for machine learning did well. And we developed our own methods that uses kernel similarity of neighbors and simple integration. It was best overall, but not always. There were cases where other methods did better, and also there were cases where using a single or subset of the omics was better than using all omics. So this begs the question of whether clustering the way we do it is the right approach, and we'll get to it in a minute. One thing that we found out in the process is that the log rank test that it is broadly used uses the asymptotic approximation of the chi-square, and on the type of data sets that we have here, a few hundred samples, the results of these approximations are too optimistic, and we actually developed, implemented a method that would use a permutation-based exact test. Moving on to another approach to this method, following this study. This is joint work of Nemo and Roy from the group. And here, what we, as I told you, we observed that sometimes not all omics behave the same. For example, here you see the results of breast cancer, and you see the three omics, and the yellow cluster seems to cluster well in microRNA and mRNA, but not in methylation. On the other hand, the purple cluster seems to be doing well everywhere. Maybe we should not include all omics in all subsets. So here is our approach, which we call Monet, and the idea is to select the omics per cluster. So a module will be a set of patients and a set of omics. So here, module beta contains these three patients, and it is only includes the blue omics. Module alpha contains these samples, and it is covered by omics 1 and 2. And we developed a method hill climbing method to search the solution space. This is an NP-hard problem. So here's the approach. Suppose this is the solution in the center. You see the solution that is, I think, this diet. The center, we see the solution, the current solution, and one thing we can do is add or remove patients from omics. So here are, or switch sample between modules. Another thing that we can do, we could either add an omic to a cluster. So here now, this beta is now both blue and red, or we can remove, so alpha is now red only. So we do this hill climbing approach, and until we are stuck, and here are the results. So we apply this to the same 10 cancer types. And what you can see here is the set of omics that were included in all the clusters of all samples. So there were relatively few where all three omics were included. So there were some that a single omic was best, and a few where a pair of omics turned out to be beneficial. So indeed, very different from the clustering that we saw before. When we look at survival, we see that our new method, Monet, performed equally well to Nemo. And when we look at the enrichment in clinical parameters, and the same, they are both Nemo and Monet are doing best. But interestingly, they are not doing best on the same set of cancers. So there is an advantage of using both. Here is one application. So here we looked at data of single cell clustering. We look specifically at expression and methylation data from mouse embryonic development. And you see here the 8 clusters that we found. For example, the first cluster was gene expression only. The last 3 clusters were both omics. There was one cluster that was DNA methylation, and only DNA methylation. And when we look at the cells, we realize that it is similar across different gene layers, and this suggests what is actually known, that even if the expression is changing, the methylation still persists at this developmental stage. So to summarize this part, we developed a new model that generalizes clustering, where the set of omics is selected for each cluster. It is equal to Nemo overall, but in terms of the types of cancers on which it is doing best, they are not the same. So our suggestion is to use this complementarily, and this begs the question whether more general models could be beneficial here. Switching gears, let me present the work that was done by Nimrod and led by Jonathan, a master student of the group. So here is the setup. When we are talking about multi-omic integration, there could be a situation where we have a single omic and multiple data sets, like we see in red, and this is a well-studied batch correction problem. There could be a situation where we have the same data set but multiple omic, like we just described. And the most general situation is where we have multiple data sets and multiple omics. And specifically, what we will focus on here is the situation where we have gene expression and DNA methylation, but we will assume that the DNA methylation and gene expression are on different data sets. So one set we have gene expression, on another data set we have DNA methylation, and we wish to integrate them. So here is how we do it. We assume that we have a set of samples for which both data sets are available. And for each gene, we look at the DNA methylation signal within plus minus 10 kb from the gene. We actually exclude the gene body and the 5' UTR sites, and for the rest, we summarize the signal and we use lasso to obtain the regression coefficient. And we pick the 2000 models with the highest R square. That this will be the result of the training. For the embedding, as I said, we have one data set with DNA methylation and another with gene expression. And we use the 2000 models that we started with in order to predict the expression from the DNA methylation for data set 1. And then we restrict the genes to those that are highly variant, both in the gene expression and in the DNA methylation. So we end up with a set of typically of a few hundred genes. And then we now have the predicted gene expression and the known gene expression from the other data set. And we embed them together into a d-dimensional space using a canonical correlation analysis. And we call this method intent. Here are the results. First result, so we looked at 11 cancer types from TCGA. And what we did here is to use the training on 10 cancer types, leaving aside the 11s, and then do the prediction on the 11s and the embedding. And to evaluate the results, since we know the matching of the patients from the DNA methylation and the gene expression, we can look at the closeness of the points representing the DNA methylation and the gene expression in the embedding. And the measure that is used in the literature for this is called the fraction of samples closer than the true match in the embedded space. And of course, the smaller this fraction, the better the results. So here are the results. We compared ourselves to Liger, Syrah, and MMDMA in several versions. And on the y-axis, we see the score, the fraction of samples closer than the true match. And we see that intent is substantially better than all others in virtually all cancers. Here is one example. So this is colon adenocarcinoma. So you see here the UMAPs of the DNA methylation and the gene expression. And here is the embedding of intent. It is very well mixed. In Liger, we see strong separation. There's also substantial mixing in MMDNA, but overall, the results of intent are better. We marked 10 random points, 10 individuals on both the expression and the methylation, and you could see that typically the two omics embeddings are near to each other overall. Here is one other task that we could do. So here we looked at 4 cancer types together, and we trained on the remaining 7 cancer types, and then did the embedding. And again, the FOSS-TTM was 2 orders of magnitude better than in the other methods. But here, since we have several cancer types, we don't only want to see mixture and closeness, but we also want to see if the methods separate between the omics, between the cancer types. So here is the mixing result, similar to what we have seen before. Again, intent is substantially better. And here, the samples are colored by the 4 cancer types, and we see that intent provides a very good separation. Actually, here MMDNA provides good separation as well, but other methods less so. So here is one sample, one final example. So here we looked at melanoma samples, and we partitioned the data set into 2 halves. We used half of the methylation signal and half for the gene expression signal. And what you see here is survival plots for the resulting clustering results. So the DNA methylation by itself does not do a good job in separating. The gene expression profiles are doing much better in terms of separation. And when we take the embedded DNA methylation profile, they are doing equally well as the original gene expression profile, even though, as I said, these are 2 disjoint set of samples. So to summarize this part, we developed a method to integrate DNA methylation and gene expression profiles from different data sets. Using combination of regression to learn the individual gene models and the CCA for embedding to a joint space. here it was crucial that we knew how to match between the DNA methylation and the particular gene. This was quite useful. However, this could be generalized to other data pairs. And we see that on our test, the advantage of using this data were very beneficial. One thing that is left to be done, the embedding phase, we are now using CCA, but there are a variety of good single omic multiple data methods. Once we do the embedding, we are back to the single omic multiple data methods, and we could apply any of these methods and see perhaps we can do even better. How am I doing? Okay, so I'm wrapping up. Skip the last story and just thank my group and thank you for listening.
Wow, many questions here. Let's start.
The second part of this story, which cancers would you say were the closest, and which ones were the furthest?
So
You looked at 11 of them. Which yeah.
Oh, so you're talking closest and furthest into, we didn't compare similarity between cluster types. We compared, in the second story, we looked at the set of common omics to each pair of cancer types. So we can look at this example, let's see. So here are in this case, we have 4 cancer types, and according to intent, the SARC and the melanoma are the closest. We haven't done this globally, but this is definitely something that can be done.
Sorry. I got a bit lost in the Monet part. So when you say that some samples, some patients cluster better or are grouped better using only one omic data or multiple omics data, is that affected by the quality of the data or the lab source of the data, or how does that happen? Is it a different subtype of cancer and that's the reason, or?
So, you know, there could be certain I don't know, patient groups that are affected more by a particular subset of data types. Maybe, I don't know, microRNA is not relevant to these particular subtypes, and therefore we don't expect a clustering. This was the reasoning. We saw empirically that sometimes it is better just to use a single omic than using all omics. So this Monet was trying to generalize this and see if there are subsets that do better.
And my last question would be, in like the last part of your story where you talk about the melanoma, I was wondering if you look into other subtypes of melanoma, like acral melanoma, that it's still the same cell type. There are some similarities, but we also know the origin is different. There's no UV light in
No,
that.
we haven't done this. I'm not sure that there are enough samples in the TCGA to look at subtypes, but we can look it up. Yeah. Thank you.
Very important. Very important work. Very important work. So in the first project, I believe it is AML, you found different groups with different survival rate. Did you try to look what is unique with respect to the group of the highest survival rate compared to the lower survival rate with respect to the DNA methylation, gene expression, and mRNA profiling.
I don't think we found something common across cancer types. We looked at some particular examples like the one that I showed for the AML, and there we could match it to known subtypes. But looking at the particular omics, no, we haven't looked at it. That's a good point.
Okay, then maybe the last question.
It's very nice. I mean, I had two quick questions, maybe one, the most important one, maybe even the last part. The regression model with methylation expression. Can you comment on how specific that is to cell types and data sets? I think I understand you learn a specific model for each data set, but is there universal relationships that you can leverage across data sets or use transfer learning? And the question is it really data set specific, or are there ways to maybe, even cross leverage public resources, other data to?
Yeah, so the way we thought about it is that this is, we expect something, we expect this relation to be universal. So if we apply our method to a subset of the cancers, then we use everything else to do the regression. It might be more powerful if we can identify the most appropriate cancer subtypes, but we haven't done this. Yeah.
Okay, so thank you very much. So we move on with the next speaker, which is Roderick, and he will be talking about the epigenetic logic of gene activation.
Yeah, I will not thank the organizers for inviting me here. I apologize because I actually breaking out a bit the topic of this session because I'm